Hello guys, welcome to Vedantu NEET English. I'm Miss Gopika, your own biology master teacher. I hope I'm audible, visible and the presentation right behind me is visible. Welcome all of you to Molecular Basis of Inheritance Part 1 NCERT line by line, which is not done very commonly, but today we are going to decode NCERT for Molecular Basis of Inheritance, which I've also taught you in RNA series, right? So if you want a lecture where there is only content of me writing, definitely type Molecular Basis of Inheritance RNA series and the video will pop up for you. Okay, so I hope I'm audible and visible. Please let me know in the chats if we are live yet. Quickly let me know and let me also refresh. Yes, we are live, right? Awesome. Okay. So good, yes. Hello, good morning, good afternoon. We're already at good afternoon right now, right? Yes, quickly share the link with your friends and ask them also to join, okay? Yes, audible, visible. Yeah, students, the camera setup has changed, so that's why I look like this. For today, just adjust, okay? We'll fix it in some time. Okay, we'll fix it in some time, right? But you can see the presentation, no? Presentation should be visible. Even if I'm not visible, it's okay, but presentation should be visible. So you can see, right? Now, molecular, okay, this is the lights. The setup has changed. I have already informed the team. They'll come and check. Okay, I'll already inform the team. They'll come and check. Don't take tension about the light. Okay, it's a dark setup. Okay, I have informed once again. Okay, done. Done. Okay, chalo, let's do. Okay, do you all know how many marks this chapter fetch in your NEET examination? You will be shocked. Okay, around 10 questions come from this chapter. Okay, 10 questions come from this chapter. That means 40 marks. Okay, 40 marks you can expect from this chapter. Students, starting only, they have board has started doing, Dhooma chale, it's moving everywhere. Okay, so today we will do NCERT. You have already seen my lectures where I teach you the entire content by notes. Today we will see how NCERT is going to be different or what are the words in NCERT. Okay, yes? Okay, chalo. Students, don't worry, it will get fixed. Don't keep complaining, it will get fixed. Okay, give 2-3 minutes. Okay, they are all coming to office, so once they come, it will get fixed. Okay, yes, let's start. See, deoxyribonucleic acid or your DNA and ribonucleic acid are two types of nucleic acids found in living systems. Yes or no? Now, what is this nucleic acid, man? So, there was a scientist called Altman. Who was the scientist? Altman. He told that this nucleic acid, first scientist was Friedrich Mischer. Okay, first scientist was Friedrich Mischer. He told that he was, students, like, as I told you, when you go to the fridge and when your mom asks you to take, green peas and come, it will be kept in an ice cream box. So you will be like, oh my god, ice cream is there. You open that, you will find green peas. That means what? By fluke, you found that it is green peas. You did not search the whole fridge. Same thing happened with Friedrich Mischer. He was working on pus cells. But by mistakenly, he found something called as nuclein. Okay, by mistakenly, he found something called as nuclein and he understood that this fellow is not something simple. This fellow is something big. Okay, this nuclein is someone so big. So, he took that nuclein and he was like, this is nuclein. This has something to do with our inheritance, something to do with our genetic information. Then another scientist came. Okay, what did that other scientist tell? Very good, very good. Another scientist told that, wait, this nuclein has a acidic nature. Nuclein has what? An acidic nature. So, why don't we call nuclein as nucleic acid? Okay, why don't we call nuclein as nucleic acid? And that is how this two nucleic acid came into existence. Is this clear now? How the introduction happened? Nuclein was found by your Friedrich Mischer, that also by fluke. And later, Altman scientists told that, wait a minute, this nuclein has a acidic nature. So why don't we call it nucleic acid? That is how your nucleic acid word came. Now here what are they telling? They are telling your deoxyribonucleic acid, that is your DNA, and RNA are the two nucleic acids present. Okay, two nucleic acids that is present on earth. Now DNA acts as a genetic material in most of the organisms. Students, 99% organism, who is the genetic material? DNA. Ma'am, then what about RNA? Is RNA ignored? Tell me, is RNA ignored? Yes, little bit it is ignored. Okay, why? Because of some reasons which you are going to study further in the class. DNA, though it also acts as a genetic material for some viruses. Give me the example of some viruses that it is going to act as a genetic material, all of you in the chats. I have taught you this many times. Okay. Scientist names, see, Altman and Friedrich Mischer, when we teach, we tell, but usually it's not mentioned as such in your NCERT. Friedrich Mischer is mentioned, so you can remember the names. Okay, it's very easy names, no? So you can remember it. Tell me, which are the, who is going to have RNA as the genetic material? They have already mentioned virus is the one who is going to have RNA as a genetic material. But can you give me some examples? Some examples who will have RNA as a genetic material? Almost all your retrovirus, right? Retrovirus, that is like your HIV. Yes or no? Human immunodeficiency virus, which causes your AIDS, is there. Then you have your tobacco mosaic virus, right? Tobacco mosaic virus. Bacteriophage has DNA. Okay, bacteriophage has DNA. You have your TMV. As well as TMV, students, retrovirus, you can remember your COVID virus. All this are going to come under the example of viruses. Okay, example of viruses who is going to have DNA, no, RNA as a genetic material. Now, mostly functions as a messenger. So, they are like, okay, you are not going to be the major genetic material, but there are some other activities that you can do. What are the other activities? RNA will act as a messenger. He will transfer some messages. He will send some messages to people, right? Not like how you send messages to your friend telling, hi, how are you? Did you have your food? Not like that. Okay, it is going to help as an mRNA. It is going to help as what, students? It is going to help as mRNA. As mRNA. Got it? Now, in this, don't worry about the notes. Your notes, beautiful notes, I have already sent it to you. Now, try to learn only NCERT line by line, because in this as such notes will not be there. Okay, all underlining portions will be there. So, you focus on trying to understand the NCERT line by line. Chalo. Guys, you all requested me for this class and I cannot see the attendance. I cannot see any of the Josh and all of that. What is this? Trusting you people and taking a class has now become a this one only, no? See, look at the attendance. They were like, ma'am, we will attend, ma'am. We will do that, this, they told in the telegram. Now I can't see any of them. Okay, chalo. Now, in some viruses, mostly functions as messenger, has additional roles as well. It functions as an adapter. It functions as what? Adapter, and a structural, and in some cases as catalytic molecule as well. So, it is going to act as an adapter. That means what? It is going to adapt to the situation. No, it is going to help, like your tRNA. It is an adapter molecule. That means mRNA has some code. Now, to read that code, I cannot call someone else, no? So, RNA is like, don't worry, I will do the job for you. I will read the code, and also at one end of my loop, I have an amino acid acceptor. I will add the amino acid acceptor and I will make sure that you get a polypeptide chain as well. So, this job also is done. Yes or no? This job also has been done. Now, in class 11, you have already learned the structure of nucleotide and the way these monomer units are linked to form nucleic acid polymers. Yes or no? Have you learned it? Now, in this chapter, we are going to discuss the structure of DNA, its replication, the process of making RNA from DNA, that is transcription, the genetic code that determines the sequence of amino acid in proteins, and the process of protein synthesis, and elementary basis of the regulation. So, what we are going to learn in this chapter? We are going to learn how DNA is formed, who is the constituents of DNA, how we can form from DNA to RNA, RNA to protein. That's all we are going to learn in this chapter. Okay, first paragraph is clear, students? Yes or no? Now, we are going to come to the most complicated parts. Okay, now complete nucleotide sequence of human genome. Okay, now what is this nucleotide, ma'am? You never spoke about nucleotide. So nucleotide is a small monomeric unit. Okay, many nucleotides together forms the polynucleotide chain, and that polynucleotide chain forms your DNA. Yes or no? So this nucleotide has like your monomeric constituents to form the polynucleotide chain. Now they will come, okay, who is going to come? They are going to come, attach and form your entire DNA molecule. DNA molecule as well as your RNA molecule. Clear or not? Now look here. During the last decade has set in a new era of genomics. In the last section, the essentials of human genome sequencing and its consequences will also be discussed. So here they are talking about your HGP. Okay, they are talking about your HGP and they are talking about your DNA fingerprinting. Okay, these two concepts also will be discussed in the end of the chapter. Okay, yes. Yes, Madhumati and Tanuja is doing their job. Okay. Yes. Now let's see. Let us begin our discussions by first understanding the structure of the most interesting molecule in living system. That is who, students? Not you, but it is the DNA. In subsequent sections, we will understand that's why it is most abundant genetic material. Most abundant genetic material means what? Most abundant genetic material means that means most of the organisms, 99% of the organism is going to have this fellow as the genetic material. That is your DNA. Okay, most of the living organisms, are going to have this fellow as the genetic material. Clear? Very nice. Okay, and we'll also understand what it has relationship with RNA. What relationship it has with RNA? RNA is going to do individually the job of messenger, right? It is going to act like a messenger. It is going to act like a catalyst. What else, students? Ma'am, adapter. And I cannot ignore the fact that it is also a genetic material. Clear? Clear or not? Okay. See, this is the situation.
So beautiful.
Students, instead of solving problem, Mithun is fixing the issue by telling me that I look fine. I know I look beautiful, but I look more beautiful than this. Fix this. My students are not able to see. They're like, ma'am, switch on the light.
Oh.
Oh. No, guys. Yes, wait, fixing. This is it better? And they'll tell, no ma'am, it's not better.
It's fine. Or kya tha? And what all things we can change?
Yeah, voice will not be visible because voice has to be audible, no?
Voice is not visible, they are telling.
My kids are very funny. Wow, Madhumati, thank you.
Abhi theek hai kya?
Now okay, no? Okay. Now students will come back, okay, to being beautiful again once this camera is fixed. No, ma'am, it's not better. Trisha, adjust Trisha. I think now if you reduce this one, it'll be better. This brightness.
Let me set it to default first.
Okay.
And then let's
No, 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 no.
Saturation and value. हाँ, समझिए.
Just see one second.
Yellowish come on.
Yeah.
Then let's increase the comma a bit
Mm-hmm. Fine, fine.
and contrast a bit. Fine.
Yeah, better. Better, no, guys? Light is there. In life, light is little less, students. That's why. Better, no? Oh, God. I have gone through like 3 shades of colors in this whole video. Guys, attendance I cannot see, okay? Attendance I cannot see. Okay, done? It's okay, adjust, okay? We'll fix the issue, okay? Now, suddenly I cannot fix the issue and I came morning, no? So, there was no one in the office as well. So, don't worry, it will be fixed. Chalo, let's start. So, we haven't come to the major portion, okay? Now, we are just going through the surface level. This happened, that happened. We are just talking about it, okay? We're just talking about it. See, DNA is a long polymer of deoxynucleotide. Now, deoxy means what, students? Ma'am, deoxy means that something, deoxy. Tell me, deoxy means, ma'am, one oxygen could have been removed from the ribose sugar. Yes or no? One oxygen is less. Okay, one oxygen is less. So, that could be the reason, okay, that could be the reason that, okay, it's called deoxyribonucleotide, okay, deoxyribonucleotide, okay, clear? Okay, done. Okay, very good. Now, the length of the DNA is usually defined, okay, look here, the length of the DNA is usually defined as the number of nucleotides or a pair of nucleotide referred to as base pair. That means, let's say this is the DNA of whose we'll take, okay, some names are very difficult to read, yeah. So, here, look at this is the DNA. Now, how do I know how long this DNA is? I have to count the number of nucleotides, yes or no? So, the length of the DNA is going to be denoted by the number of nucleotide. Or this is single, okay, this nucleotide is single. Now, we have to find the base pairs. Base pairs means what, students? 2 people together, no? Now, adenine is there. Adenine will bind with thymine. So, this is a base pair, 2 people together. So, you can neither count the number of nucleotide individually if it is a single strand, or you have to count the base pairs. 2 people are there, so we will count number of base pairs to understand how long, okay, what is the length of the DNA, okay, what is the length of the DNA, is this clear? This is also a characteristic of an organism. That means to organism it can change. Okay, now honeybee and we will not be the same. Same way a bacteriophage and us will not be the same, like that. The number will change. So, look here, for example, okay, here they have given a bacteriophage known as 5174, okay, has 5386 nucleotides. I have told you this before also. Don't ask me trick for this. I cannot find a trick for this, okay. Many people do some reduction in numbers and all, but I feel it's very difficult, okay, because you are already weak in maths, right. So, now upon that if I put some numbers also, you'll be like, ma'am, everything is going in space, okay. Solar system I'll have to see above your head like this. So, that's why I'm not playing with numbers. I understand how difficult maths can be for all of you. So, let's not do this, okay, let's not do this. Yes, who is this? Yes, so look here. So, here your bacteriophage 5174, 5386 nucleotides. So, students, since here they have mentioned only nucleotide, that means this is a single stranded structure. Yes or no? Single hai? No, single stranded structure it is. That is why they are mentioning the word nucleotides. They are not mentioning the word base pairs. If they mention the word base pairs, that means 2 strands are there. 2 strands are there. Okay, here also one person is there, here also is there. They both are bound with hydrogen bonds. Okay, now bacteriophage lambda, okay, lambda has 48502 base pairs. That means here they are not single, here they are double stranded structure. Clear or not? This is just a paragraph, okay, just a topic, like, you know, where you have to know the number, yes. Oh, full, they are praising each other. E. coli, that is Escherichia coli, has 4.6 into 10 to the power 6 base pairs. Who? E. coli. Your one and only E. coli that you have been studying so much now. E. coli has how much? 4.6 into 10 to the power 6 base pairs. Now, and a haploid content. Okay, haploid content of human DNA is going to have 3.3 into 10 to the power 9 base pairs, but here they are talking about the haploid content. Okay, 1 second, students. Here they are talking about the haploid content. They can ask you question on the diploid content. If they ask you a question on the diploid content, how are you going to answer? Tell me this in the chat. I have taught you this before also. If it is going to be about the diploid content, then you're just going to do 3.3 into 10 to the power 9 into 2. Okay, into 2. So, if we do this, okay, this you're going to do into 2. How much is the answer, students? Into 2 you are going to do, right? So, you will get 6.6, okay, approximately 6.6, right? So, you have to do some multiplication, okay, but right now I am just directly going to write, it is going to be 6.6. So, 6.6 is going to be of the diploid content. Haploid content, where it is going to be there? It could be seen in the sperm, it could be seen in the egg. Other places you do not see the diploid content, right? Otherwise, other places you do not see the haploid content. So, in few places in our body, in few, specific gametes, we will be seeing the haploid content, right? Very good, very good. Awesome, awesome, awesome. So far good? So far clear? Yes or no? All of you in the chats? So far clear? Yes, very good. No green hearts, red hearts. Yes. Okay, very good. Now, let us discuss the structure of the long polymer. Now, what we are going to learn? Now we are going to learn the main portion of this chapter, okay? I want all of you for next 5 minutes, don't talk to each other, just try to understand how the bonding is happening. Only this part is tricky in this chapter, students. Rest everything is very, like, it's very beautiful to understand, okay, that this much things are happening in our body. So next 5 minutes, full focus, okay, full focus, no talking with each other, listen how the bonding is going to happen, and I will make sure that you know every single line, every single line. See, look here. Let us recapitulate the chemical structure of a polynucleotide chain, that is your DNA or RNA. A nucleotide, okay, they are telling that the nucleotide which I spoke about before has 3 components, okay. Nucleotide has 3 components, they are telling. What are the components? A nitrogenous base, okay, a pentose sugar, okay, ribose in case of RNA, that we will come into, and deoxyribose for DNA, and a phosphate group. So they are basically telling that the nucleotide has 3 components, okay. Many nucleotides together forms the polynucleotide chain. It's simple, no? From the word itself you understand, poly means many, nucleotide means that has 3 components. What are the 3 components of a nucleotide? First component of a nucleotide they are talking about is a nitrogenous base. Okay, nitrogenous base. This nitrogenous base will have 2 content, which is your purines and pyrimidines. Okay, they will have purines and they will have pyrimidines. Purines and pyrimidines. First word is clear. Nitrogenous base is clear. Okay, next word that they have spoke is about a sugar. So they are telling in the case of RNA, it is going to be a ribose sugar. Okay, shall I take a slide and teach you this? Okay, wait 1 second. Yeah. Okay. So students, here I am taking a slide and teaching you this. They are telling that in the case of your nucleotide, okay, nucleotide, they are telling 3 components are there. First component that they spoke about was a nitrogen base or nitrogenous base. In that nitrogenous base, I told you that there are 2 components, okay, which we are going to discuss later in the portion. So first you understand the general idea, then I will take you back to NCERT. Nitrogenous base, what are they? Purines, ma'am, and pyrimidines, ma'am. Purines and pyrimidines, right? In this purine, students, very carefully listen to me. It is a 9-ringed structure. What ring structure? 9 ringed structure. Got it? 9-ring structure. And in this 9-ring structure, there are portions where nitrogen is going to be present, and that is seen where and all? In the first, third, 7th and 9th position. Clear? It is a 9-ringed structure. The nitrogen is present in the first, third, 7th and 9th position. Clear? So, I am writing it here. Super clear so far? Yes or no? Let me know. Nitrogen is present. Okay, ma'am, understood. Now, ma'am, tell me about pyrimidines. From the name, students, purine, chuttu name, no? But it has 9-ringed. Pyrimidine, long name, but it has very short things. That is, this is only a 6-ringed structure. It is only a 6-ringed structure. And in the 6-ring structure, nitrogen is going to be present on the first and the third position. Because small ring, no? So, we cannot have 4 positions. We are only going to have 1 and 3 positions. Now, if you see the structures, students, you are going to, okay, this is one structure. Okay, this is the purine. Okay, this is the purine and this is going to be a pyrimidine. This is a 6-ring structure. This is a 9-ring structure. Can you look at it and understand? Yes or no? Tell me this. Okay, so if we start labeling 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, this is one structure. Then if you start labeling 1, 2, 3, 4, 5, 6. Okay, this is a 6-ring structure. In that I told you that nitrogen is present on the first. Okay, nitrogen is present on the first. Nitrogen is present on the third, it's present on the 7th and it is present on the 9th position. Here it is going to be present in the first and in the third position. And students, is this a heterocyclic? Is it heterocyclic? Let me know in the chats. This question has come, so you should be aware. Is it heterocyclic or not? Yes, ma'am, it is heterocyclic. Okay. If they ask you the question, who is heterocyclic, you should tell nitrogen base is a heterocyclic. Now, the second component that they spoke about was the sugar. Yes or no? Second component that they spoke about was the sugar. Now, I am telling just the general idea. I will tell you more about adenine, guanine, cytosine and all of that. Okay. Yes. Heterocyclic. Okay. Very good. Tell me the doubt. Okay. Now, sugar, they are telling that, ma'am, 2 things are there in sugar as well. One is going to be a ribose sugar, okay, and one is going to be a deoxyribose sugar. A deoxyribose sugar. Sugar molecule with normal sugar molecule, no. Now, ribose sugar diagram is going to be like this. Okay, this is the ribose sugar. It's going to be OH, H, OH, H, H, OH, CH2OH and H. Okay, this is a ribose sugar. Got it? This is a ribose sugar. Got it? Now, deoxyribose means what? Deoxyribose means what, students? That means, ma'am, one oxygen somewhere went missing. Okay, some oxygen somewhere went missing. Okay, so here also I am drawing OH, H. Here in the second position I am only putting a H. Now, when I move, you will see this. Okay, 1 second. CH2OH, H. Tell me which carbon I made a change. In which carbon among this I made a change? Can you all tell me in the chat? Ma'am, between this, can you identify which carbon there is a change? Is it in the, so if you want the naming, let's do the naming as well. So, this is going to be a 1 dash, okay, 2 dash, 3 dash, 4 dash, okay, and this is going to be a 5 dash carbon. Same thing goes here, 1 dash, 2 dash, 3 dash, 4 dash, okay, and 5 dash. Tell me in which carbon did I make a change? Very good, Trisha, very good Sharan, very good Suharshita. Yes, guys, all of you, mark your attendance quickly. Minecraft, Tanuja, Madhumati, very nice. Okay, in the second position, ma'am, you did not draw an O in the DNA. Okay, this ribose is going to be a part of your RNA, because what is the full form of RNA? Ribose, right? Ribonucleic acid. So ribose means this is the ribose. Then here DNA, it is deoxyribose. That means this is going to be coming in the DNA. And this one OH which is missing, or this one O, one oxygen which is missing in the 2' end of your DNA, is making DNA so capable. Okay, DNA so capable. So this is clear, general idea is clear. Now if we go further, right, if we go further, students, we will understand that purines have two people. Purines have two people. What are they? Adenine and guanine. You can remember pure as gold. Okay, pure as gold. Okay, PAG, pure as gold. In pyrimidines there are three. Okay, in pyrimidines there are three. That is going to be cut. Okay, it's going to be called cut. C stands for cytosine, U stands for uracil, T stands for thymine. T stands for thymine, cut. In this, uracil is going to be present only in your RNA. Okay, it's going to be present only in your RNA. Clear so far? Yes or no? This is clear, ma'am. This is the general idea of what that two lines in your DNA, I mean in your NCERT, it was mentioned. Now next we will learn about bonding. Before that, let's go back to NCERT. Okay, let's go back to NCERT here. Okay. So this way we are going to understand every single line of NCERT. Okay. Now look here. A nucleotide has three components. Okay, ma'am. A nitrogenous base, a pentose sugar, ribose in case of RNA, deoxyribose in case of DNA. My students already know that on the second carbon the O was missing. That is why they are mentioning ribose and deoxyribose. Now there was a phosphate group. Now students, this phosphate group is nothing. Okay, this phosphate group, it is nothing but your PO4 3 minus. Okay, PO4 3 minus. That is your phosphate group. Luckily, you do not have to learn a lot of diagrams for this or any structures for this. Now, there are two types of nitrogenous bases. What are they? Purines and pyrimidines. So, under purines, you are going to see adenine and guanine. Okay, and under pyrimidines, you are going to see, under pyrimidine, you are going to see cytosine and uracil and thymine. That is your cut. Okay, CUT, cut. Cut is going to happen. Cut. Okay. Okay. Yes. Now cytosine, uracil and thymine. Now cytosine is common for both DNA and RNA, as I told you. Uracil is the only one that you are going to see in RNA. Thymine and cytosine both are going to be present in your DNA. Cytosine is going to be present both in DNA as well as RNA. Okay, DNA as well as RNA. Clear or not? This is thymine is going to be present only in DNA. Uracil is only going to be present in your RNA. Understood, ma'am? Okay, understood? Any doubts? Now, a nitrogenous base is linked. Listen to me carefully. Okay, a nitrogenous base is linked to the OH of the first dash, that is 1-C pentose sugar. Students, what do they say? They are telling that your base, nitrogenous base, is going to be linked to the first carbon of your sugar molecule. Can you relate it? Yes, ma'am, we can relate it, right? That one, okay, first carbon, to that first carbon, a base is going to be linked. Now, let's say if I draw the structure here, what are they talking about? They are telling that this is your sugar. Okay, this is your sugar. Yes, to this sugar, I'll label this. Students, now I am drawing a ribose sugar. Okay, you can look at the diagram and understand. No? Oh, I'm so sorry. It's an alarm. It's my DNA alarm. Okay, done. Okay, so students, here it is going to be CH2OH, okay, and H. They are telling to the first carbon, okay, to the first carbon there is going to be a base that is going to attach. It can be any base, it can be a purine or it can be a pyrimidine. Is this clear to all of you? To the first OH, okay, first carbon of your sugar, base is going to attach through what linkage? This linkage is called as N-glycosidic linkage. N-glycosidic linkage. If you learn this paragraph, let me tell you, everything else in this chapter is going to be very, very smooth. Okay? Very, very smooth. Clear or not, all of you? Tell me. Okay, done. Done. Now, through a N-glycosidic linkage. So, this is the sugar, this is the base, and this base is attached to the first carbon of the sugar, and this bond is called as N-glycosidic linkage, and this linkage is going to end up forming what for us? A nucleoside. So, I have always told you this. If it is a nucleoside, whom are we pushing to the side? All of you tell me in the chat box. If we are going to talk about nucleoside, whom are we pushing to a side? One person we are pushing, telling that, hey, I don't want you. Okay, we both will make nucleoside, we don't want you. If you come, we will become nucleotide. I don't want to form tide, I want to be side. Who is that? Quickly in the chat. Quickly in the chat, let me know. Who is that tide? Side. Who is going to be pushed to the side? Quick, quick, quick. Very good. Very, 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 very good. Okay. 1 second, students. Yeah. Awesome, awesome. Yes. Yes. Okay. Yeah, it's like that only. So, nucleoside means you are pushing phosphate to one side. Okay. Phosphate is not there. Okay, phosphate is not there. You're pushing phosphate to one side and telling that, hey, you know what, I don't want you to be here. Okay, I don't want you here. Sugar and base will form the nucleoside. Now students, such as, so whenever it is a nucleoside, the suffix is going to be "-ine". What is it going to be? The suffix is going to be "-ine". And if it is a nucleotide, okay, if it is a all this I have written the notes and given it to you on the Telegram channel. So quickly go and check the Telegram channel. You can join the Telegram channel, Vedantu NEET English Telegram channel, where I have shared all these new notes handwritten. Now I am teaching you through NCERT because we are doing a revision. Okay, but I have written all of this and given the notes on both Biology by Gopika as well as on Vedantu NEET English. So go check it out, and if you want the notes, just let me know. I will reshare it again today. Okay, chalo. So, I told that if it is a nucleoside, "-ine" is going to be the suffix. So, can you see here? Adenosine, deoxyadenosine. Okay, clear? Sine is coming. Okay, "-ine" is coming. Here you see guanosine. Okay, guanosine, deoxyguanosine. Now, students, guanosine is going to be present in your RNA, right? Deoxy is going to be present in your DNA. Understood that? Same way, cytidine. Cytidine is going to be present both. It's a nucleoside. It's going to be present both in DNA as well as RNA. No, same way. See here, this cytidine is going to be present in RNA. Deoxycytidine, DNA. Same way, uridine. Uridine is only going to be present in your RNA. Only going to be present in your RNA. Understood? Understood? Uridine is going to be present in your RNA, and deoxythymidine is going to be present in your DNA. So, if you write, okay, kuti, if you write above your book itself, on your NCERT itself, write DNA, RNA, write DNA, RNA, you will be sorted. You will understand. You will remember all the time. Okay, this is there in DNA, this is there in RNA. Understood? So, whenever it is a nucleoside, the suffix is going to be "-ine". Can you see? "-ine". If it is going to be a nucleotide, the suffix can be "-ate", "-ic acid", it can be anyone among this. Now look here, same thing goes here. When a phosphate group is linked to the OH of the fifth carbon, so they are telling that to this carbon, there is going to be a PO4 3 minus that is going to be attached to the fifth carbon. This is the fifth carbon. Yes, ma'am, it is the fifth carbon. I'll write it again neatly. Okay, I'll write it again neatly. Evolution part 1 notes. Okay, I'll just check that. When a phosphate group is linked to the OH of the fifth carbon of a nucleoside through a phosphoester linkage, okay, phosphoester linkage, a corresponding nucleotide or a deoxynucleotide depending upon the type of the sugar present is formed. Did you understand English? Yes or no? Did you understand the English? Tell me in the chats, all of you. Understood? Let me see who understood this English. All of you understood? No, right? So I will explain it again to all of you. Look here, what they are talking about, students. They are telling that, now we understood the general idea, yes or no? In this slide we understood what is the general idea, how the structure is looking. Now we will learn about the bond. Okay, now we will learn about the bond. So they are telling that bonding is going to happen. Between whom and whom? Between all of these people. Okay, so generally if it is a sugar, okay, plus a base, what will it form? All of you tell me in the chats. If it is a sugar and if it is a base, what will it form? Yes, doing it once again. So, sugar and base is going to form nucleoside. Okay, nucleoside. I already mentioned this to you. Now, I told you that the suffix for nucleoside is going to be "-ine". Okay, so like that we have adenosine, guanosine. This is for ribose sugar I am writing. With deoxy, it is going to be for your DNA. Okay, deoxy, it is going to be for your DNA. Now, guanosine, adenosine we have. Now, deoxythymidine, only present in DNA. Like that, uridine, only present in your RNA. Okay, only present in your RNA. Like this, understood? No, "-ine". I just wrote the example now. Now, they are telling that when we attach a phosphate to this, okay, when we add a phosphate to this, that is sugar plus base plus phosphate is going to form what? They are telling me that it is going to form a nucleotide. It is going to form a nucleotide. Understood? Tide. Okay, and what is going to be the suffix for this? It can be ate or isic acid. Okay, it can be ate or isic acid. So, nucleotide will be formed. Now, generally, I am going to write here. Okay, generally, I am going to write here and then I will show you with the diagram as well. So, students, here, where do I write here? Okay, they are telling that whenever a sugar, okay, binds with a base, okay, the binding is going to happen with the help of the bond. The bond is going to be N-glycosidic linkage. N-glycosidic linkage. Understood? Now I have written very generally. Okay, I have not drawn any diagram. I have not confused anywhere. Just see sugar plus base if they have to bind with each other. Okay, that means imagine two Malayalis are getting married. They will get married in what? In Kerala. Okay, example I am telling. Same way, two Tamilians are getting married. They will get married in Chennai. Two Telugu people are getting married. Okay, they will get married in Andhra Pradesh. Two Kannadigas are getting married. They will get married in Karnataka. Like that, no? So, two people have to, if they are of the same state, if they are of the same culture, if they are of the same language, they will get married in a specific temple in their area. Same way here also, if sugar and base has to bond, it is going to always bind with an N-glycosidic linkage. Okay, now let's draw the diagram and understand. Let's draw the diagram and understand. So, I am going to first draw a sugar. Okay, I am going to draw a sugar. I will draw a ribose sugar. Okay, I will draw a ribose sugar. Ribose sugar means when I am telling that only you should remember, okay, ma'am is talking about RNA. Okay, this is a sugar. Okay, this is a sugar. They are telling that whenever a sugar is going to bind, okay, whenever a sugar is going to bind, it will bind on the first carbon. Okay, it will bind on the first carbon and this is going to be a base. Okay, this is going to be a base. Now, that binding, okay, that binding, what is going to happen? It's okay. Tanuja and Madhumati, let's forget about it. Okay, let's forget about it. Okay, no more target for today. Okay, we are going to focus on understanding, right? So, here students, look here. This binding is going to happen because of what? Ma'am, because of N-glycosidic linkage. Anyone has doubt in this? Anyone has doubt in this? I am teaching very slow. I am teaching this binding very slow. Okay, clear to all of you? It's okay guys, come, let's get back to the class. Okay, you shouldn't lose your marks for other people. Okay, tell me any doubt in this? This is a sugar. Okay, this is a ribose sugar. to be specific, this is a ribose sugar, because on the second carbon you will see an OH. Okay, this is a ribose sugar. It has bound with any base. Okay, any base, and it will bind with an N-glycosidic linkage. Done? Okay, clear, oh ma'am. Okay, then now they are telling that this can also bind with the 5' carbon, and that 5' carbon is going to bind with a phosphate. So students, this is going to be your first carbon. Okay, I have also mentioned you why they are putting this dash. See, for carbon we always put this dash. Why? Because even the purines and pyrimidines, see, this is a purine. Okay, this is a purine. Purine also I am labeling how 1, 2, 3, 4, 5 I am labeling. If I label same thing for sugar, then I will not know who is binding with whom, what is the number. To make that differentiation, we put a dash like this. Okay, this is actually they teach us in PhD and all. So that you just remember. Okay, that you just remember. Okay, very good. Now this is clear. Now they are telling to the 5th carbon. This is the 5th carbon. Agreed, all of you? On the sugar, this is the 5th carbon. To the 5th carbon, your phosphate is going to bind. Okay, to the 5th carbon. Okay, to the 5th carbon, your phosphate is going to bind. That is PO4 3 minus. PO4 3 minus. This is the binding. Okay, this is the binding. So to make this clear, I am going to erase this. Okay, to make this clear, I am going to erase all of this. I'll write it in the other side, don't worry. Okay, by the time you people copy this down, copy this bonding. Okay, I'm writing it here for you guys again. Okay, done. This bonding is clear? Yes, very good. Now they are telling that, ma'am, this bonding with the 5th carbon is going to happen because of a phosphoester bond or a phosphoester linkage. Clear to all of you? So, 2 bonds you have studied right now. That is, when a base is binding with the sugar on the first carbon, it is going to be an N-glycosidic linkage, and on the 5th carbon, on the sugar, okay, the 5th carbon is going to bind with the phosphate with the help of a phosphoester linkage. Clear to all of you? Now, they are telling one more line in your NCERT, and that line, to understand that line, let's take another carbon. Okay, let's take another sugar molecule, students. The sugar molecule is going to be, how much chemistry I have to write for you, you see. Being a biology teacher, I have to learn so much chemistry for you guys. Again, I am drawing a ribose only, okay, because I don't want to change. They are telling that a phosphate will bind with the next sugar. Okay, what happened here? How do I ask doubts? You have to put it in the comments, dear. There's no other option as of now. You have to put it in the comments, okay? Because Telegram I cannot open for doubts. Right, you can put it in the comments. I usually check all the comments of all my videos, even previous videos I check the comments. Okay, so look here, they are telling that this phosphate is going to connect with the next sugar on the 3rd carbon. Students, one more bonding is going to happen. This is going to be in the 3rd carbon. Understood? Clear or not? Yes, this is clear. This bonding also is happening. Okay, this bonding is also happening because of a phosphoester linkage. This is also happening because of a phosphoester linkage. Tell me who did not understand this? Anyone who did not understand this? Instagram also you can ask me. Biology by Gopika, Instagram also you can ask me. I do not check. I'm not very, I do not use that much of Instagram, social media like that. Okay, so I will check, but I will check a little late. Okay, because I have too much work is there, students. So look here, this phosphate is going to, this is the phosphate of this fellow. Okay, this is one nucleotide, this whole thing. This phosphate is going to connect to the next sugar. This is another sugar in the 3' end, in the 3' end with a phosphoester bond. Now together they are telling that if 2 nucleotides are combining together, we can call this 2 bonds, right, this whole thing, okay, this 2 bonds. 2 bonds means what? This one and this one as a phospho. Should I write it that side so you understand that I'm talking about this? Okay. this as a phosphodiester bond. Phosphodiester bond. Clear? Clear? All of you in the chats, I want to see fire emojis. Yes. If it is clear, put thumbs up or fire emojis in the chat. Quickly, look at the whole thing. Tell me if you understood, or else I'm ready to repeat it again for you people, because this is a solid class only for my NEET English students, for my Vedantu NEET English students. My purpose is to make them remember everything. Okay, my purpose is not like, okay, this video I have to make it very crisp, I should not talk to the students so that many people can watch it later. No, this purpose is to make you understand every single point thoroughly. Okay, I should feel like I'm your, offline teacher. I'm sitting next to you and I'm trying to make you understand. Clear? All of you. Is there one student also? Do not hesitate. Ask me, ma'am, please repeat. Okay? Okay, we'll do a quick recap. Okay, others also just revise it quickly. Okay, so students, very simple. Only this topic is going to be little hard here. The rest everything is going to be super duper easy. See, bond is going to happen. Sugar and base, it is going to form a nucleoside. That means we pushed phosphate to one side. If it is a nucleoside, we are going to use the suffix "ine." Okay, so uridine, adenosine, guanosine, like that, ine groups we are going to form. Then, if it is a sugar plus base plus a phosphate, it's going to be a nucleotide. They have already told us that a nucleotide has 3 components, right? And if it is a nucleotide, it can be 8 or isoic acid. Now here is the bonding. Bonding has to be clear for you. This is a sugar molecule and this is the ribose sugar. If a ribose sugar wants to bind with the base, it will bind on the first carbon. Okay, it will bind on the first carbon, and I have taught you this more in depth before when I taught you about how it is going to bind with the pyrimidine. Pyrimidine, I told you it will bind with the 1-9. Okay, I have taught you this also. Remember, this combination also I have taught you, right? So it will bind with a base. It can be any base right now, with a base with an N-glycosidic linkage. Now, if a phosphate, because an entire nucleotide has to be there, this is only a nucleoside. Now, if a phosphate comes and attach, it will attach to the 5' carbon on the ribose sugar. That bond is called as phosphoester linkage. Now, this phosphate will go connect with the next sugar, because we have to form a polynucleotide chain. We just don't want one nucleotide standing, right? We will form a polynucleotide chain. So, this phosphate will go and bind with another sugar molecule in the 3' end. Okay, 3' end, and that also is going to be with the help of a phosphoester linkage. So when we combine both of this phosphoester linkage, we can call it phosphodiester linkage. Understood? This is the 2 bondings that you have to study. Okay, this is the 2 bondings that you have to study. Does it feel like I'm running out of oxygen? No, students, little is there. Clear? No doubts? Everything is clear? Yes or no? 6 member, 9 member? I hope that is also clear. Okay, chalo. Now students, adenine and thymine and cytosine and guanosine, they have told, no? That also we have to learn, right? That also we have to learn. So these 2 slides are very, very important for you. When I am sharing this, I will share this also with it. Okay, share this also with it. Because Trisha, we need an OH group to go bind. That OH group is going to be present in the 3' end, right? Because in the 2' end it cannot bind. It will go and bind with the 3' end, and again the next phosphate, the phosphate of this fellow, this group also should come and bind. No, that fellow will come and bind here. Is this clear? This 5' end is not going to be free. 3' end is going to be free. That is how students see here 3' end is free, here 5' end is going to be free. That's how we tell 3' to 5'. Okay, and on the next strand it is going to be 5' to 3'. That's how we tell they are anti-parallel. Polarity is opposite. Okay, polarity is opposite. Clear? So when the 3' group is free, we tell 3' to 5'. Okay, and then 5' to 3'. We are going to come to that. Don't worry. Now I wanted to tell you guys this. Okay, Now students, adenine, okay, adenine will always bind with thymine with the 2 hydrogen bonds, okay, and guanine will always bind with the 3 hydrogen bonds. So how will you remember this? At Gopika Ma'am's class, okay. Now with you keep your respect inside your heart, okay, whenever you're telling this, add ma'am, okay, add ma'am. So at Gopika Ma'am's class, don't tell at Gopika's class, okay, at Gopika Ma'am's class. So ma'am is not there. AT means adenine and thymine, GC means guanine and cytosine, okay. Now AT will bind with 2 hydrogen bonds, and GC will bind with 3 hydrogen bonds. Clear or not? Clear? Okay, done. Very good, very good. I told you guys, no, it will not come again. I promised you guys last time itself. Okay, chalo, let's continue students. Here a little more explanation they have given. So let's see that. 2 nucleotides. So this is why I am telling you, if you read NCERT blindly, you will not understand a word. Did you understand anything? See, this 4 lines had so much content. This 4 lines had so much content. But if you would have read it alone without a teacher's help, you would not understand this. Okay, that is why always follow. Okay, now see, 2 nucleotides are linked through. See, look here. 2 nucleotides. Remember what I taught you. Are linked through 3'5' phosphoester linkage. Clear to all of you? Where is that phosphate going? From the 5' and it was linked, it is going to the 3' end. Okay. To form a dinucleotide. More nucleotides can be joined in such manner to form a polynucleotide chain. A polymer thus formed has at one end a free, listen carefully, at one end it is going to have a free phosphate entity, okay, a phosphate group, that, and at the 5' end of the sugar. Okay, at the 5' end of the sugar. That means what are they talking, students? Here, let's say if this phosphate wasn't there, okay, this phosphate, here if I attach a phosphate, let's say here, if I attach a phosphate, isn't this phosphate free? Isn't that phosphate free? Yes, ma'am, that phosphate is going to be free. Why? Because let's say this is the phosphate at the end of this nucleotide chain. That phosphate is going to be free because it's not going to connect with anyone. It is the end of that chain. So here I will call it 5' end because this end is going to be free. Okay. And this side one OH is going to be free because that is the end. So that will be called as the 3' end. Students, it's very simple. Just open your mind and try to understand. Here this phosphate is not going to connect with the next sugar. This is the end of the chain. I stop the chain here. This is the end of the chain. So this end is going to be free. So this end is going to be called the 5' end. Okay. Same way, here you see this 3' is free because no one is going to come and attach here. This 3' is free. So that will be called as the 3' end. Clear? Okay. Now look here. Phosphate moiety at the 5' end of the sugar, which is referred to at the 5' end, okay, of the polynucleotide chain. Similarly, on the other end of the polymer, the sugar has a free 3OH group. Understood? OH on the 3' end. Okay? OH on the 3' end. Group which is referred to as the 3' end. Is this clear? Please let me know in the chats. Now, the backbone of the polynucleotide chain is formed due to the sugar and phosphate. Yes or no? Sugar and phosphate is going to make the chain. Then, ma'am, what about the nucleotide? So, students, this is your DNA. Okay? This is your DNA. One strand and another strand. This became like that sugar-free ad. Okay. Now see, these two are formed. These 2 strands are formed because of the sugar and phosphate. But at the same time, your purines and pyrimidines are going to project inwards and they are going to make sure that these people are going to stay in place and the bonds are going to be stronger. Clear? Understood? They are going to project inwards and they are going to be sugar. I mean, they are going to be stronger. It was sugar. Okay. The nitrogenous base linked to the sugar project from the backbone. Dun, 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 dun. Okay. We'll continue. Chalo. DNA as an acidic substance present in nucleus. See, this is what I told you. Frederick Misher is mentioned here. Look here, DNA as an acidic substance present in the nucleus was first identified by Frederick Misher in 1869. Okay, in 1869. He named it as nuclein. Okay, he named it as nuclein. However, due to technical limitation in isolating such a long polymer intact, the elucidation of structure of DNA remained elusive for a very long period of time. That means what? Students, they were not able to identify this long polymer, no? Imagine many nucleotides are coming together and making a big polynucleotide chain. So, they were not able to understand what is the structure of this? How does this actually look? How is this? Okay, we understood that the bond is happening, but we're not able to understand how they are going to look like. Okay, are they going to be like this, or are they going to be like this? What is happening, Ratis? For you, everything will be missing. Your dictionary is not even Oxford. People will buy your dictionary. Okay. You're that famous, no, in spellings. Okay, so however, due to technical limitation in isolating such a long polymer intact, the structure of DNA still remained, unclear. Okay, so here it was only in 1953 that James Watson and Francis Crick, based on the X-ray diffraction, they understood that DNA is actually a double helical structure. That means two strands are there. Okay, two strands are there like this, and those strands in the between the nitrogenous bases are projecting inwards. Clear? Yes. Now, James Watson and Francis Crick, based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin. In this, how many people got, awarded, students? James Watson, Crick and Wilkins got awarded, but Franklin did not get awarded because it was after her death. Okay, proposed a very simple but a famous double helical model for the structure of DNA. One of the hallmarks of their proposition was base pairing between the two strands of the nucleotide chain. So, what did they prove? Okay. Both the telegrams you can join. Okay, Vedantu NEET English telegram as well as biology by Gopika. On both the telegrams, I usually share the notes. So, what did they do? They did X-ray diffraction. Okay, they did X-ray diffraction and they noticed the double helical structure. They noticed the double helical structure. Clear? Clear, thumbs up. Okay. Now, one of the hallmarks of their proposition was base pairing. Base pairing between the two strands of the polynucleotide chain. However, this proposition was also based on the observation of Erwin Chargaff. That's how, students, what rule came? Chargaff rule came, ma'am. That is where the ratio of adenine and thymine and the ratio of guanosine and cytosine are constant and equal to 1. What are they talking about, students? They are telling that how many adenines are there? That many thymines should be there. How many cytosines are there? That many guanines should be there. Is this clear to all of you? Yes or no? Okay. So, what did they say? They called it the Chargaff rule. This rule is very important for you. They will give you problems based on this, and I think you can do this problem once I teach this. Okay, I'll give you, so in that 2 minutes you will do the problem. Okay, chalo. Now look here, they are telling that how many purines are there, that many pyrimidines should be there. Oh, so they are telling ratio of purine is equal to the ratio of pyrimidine. Clear to all of you? Ratio of purines is equal to the ratio of pyrimidines. That means how many adenines are there? That many thymines should be there. How many guanine is there? That many cytosine should be there. Correct or not? If pairing has to happen, let's say 10 people are there in the class, I am telling make a group of 5. Make a group of 2 and you will easily make 5 groups. Correct? Now if I tell you 10 people are there, I am telling make 3 people each in a group, one person will be left out. So when I am doing that, both the numbers should be equal. Both the numbers should be equal. That means how many adenines are there, that many thymines should be there. How many guanines are there, that many cytosines should be there. Clear or not, all of you? So this will make sure that the structure of DNA remains properly and the double helical structure is proper. Yes, clear? Okay, very good. So adenine, oh god, my finger also is writing for you guys. Okay. Okay. How many adenines are there? Adenine is equal to thymine by, okay, guanine is equal to cytosine. But let me ask you guys a question. Okay, I'll ask you a question. All of you will answer. Is it necessary? Okay, this question is necessary. Yeah, question is necessary. Is it necessary that AT is equal to GC? That means adenine and thymine should always be equal to GC. Tell me in the chats. Tell me in the chat box. Yes. Okay. Tell me the ratio of adenine thymine combination and guanine cytosine combination, should they have to be equal? Charan is telling no. Okay, other people, Harshan is telling yes. Okay, let me see more answers. More answers? No ma'am, yes ma'am. No ma'am. Okay, Harshan's, it wasn't Harshan's answer. Okay. Nice. Okay. Very nice, very nice, awesome. You people are brilliant. No ma'am, they don't have to be equal. Why will they be equal? These combinations don't have to be equal to 1. There are regions on our DNA itself where we have AT rich region and we have GC rich region. So it's not necessary that this combination has to be equal, but this has to be equal. Okay, now all of you do a numerical for me. Okay, all of you do a numerical. So in a DNA, okay, in a DNA, they found that adenine is equal to 30%. Okay, now I want you to calculate thymine, guanine and cytosine. Okay, I want you to calculate thymine, guanine and cytosine, or just calculate thymine. Calculate thymine, cytosine, I think all you can calculate, okay, and guanine. Yes, do this question for me, all of you, quickly in the chats. Yes. More difficult maths, sir. I'll hit you people. Okay, T is 30, you guys are telling. Okay, what about C and G? Very good, Maharaj. Very good, very good, very good. Akshaya, very good. Madhumati, Medico, Suharshita, Yashu, Harshain, Tanuja, Charan, Akshaya, Trisha, Selvi, Yashu, very nice, very nice. Awesome. Ma'am, how did you guys find it? How did you guys find it? Ma'am, it's so simple. You already told us that if adenine and thymine should always be equal. So, let's say 100% is the total, no? So, they have told that adenine is 30%. So, obviously, guanine also will be that, right? Sorry, obviously, thymine also will be that, right? Thymine also will be 30. If these two are 30, that means they both will constitute, that is purine is going to constitute a total of 60%, right? Then how much will be left? Ma'am, 40% will be left. In that 40%, cytosine will be 20%, because if cytosine is 20%, then guanine also will be 20%. Correct? Guanine also will be 20%. Yes, very good, very good. Is this clear? This is how you're going to calculate. Okay, this kind of questions have come before also. So, these questions will keep coming. Okay, clear? This Chargaff rule was clear? Awesome. Now, we will go back to NCERT. See, the base pairing confers a very unique property to polynucleotide chain. They are said to be complementary to each other. Okay, they are said to be complementary to each other, and therefore, if the sequence of bases in one strand is known, then the sequence of the other strand can be predicted. Yes or no? Yes or no? Tell me the answer for this. That is, if I am asking you guys that I am giving you this strand. Okay, I am giving you this strand which is from 5 dash to 3 dash. Okay, and the strand is A, T, G, A, C, C, T. Can you all tell me what is going to be the other strand? Tell me the other strand. 3 dash to 5 dash strand, you people will give me the sequence. Quickly, quickly. Give me the sequence so I can take 2 minutes break sitting. Take your time. Thank you, Akash. See, it is A, T, G, A, C, C, T. I think it's easy for you guys, right? Very good, very good. Ma'am, I have the answer for it. Right, I have the answer. What is going to be the answer? I know that A will always bind with T, T will always bind with A, G will always bind with C, A will always bind with T, T, G, G, A. Correct? So, this is the combination. So, that was the statement that was mentioned, that if I know the sequence, okay, if I know the sequence of one strand, right, I will be able to find the other strand. Okay, I'll be able to find the other strand. Okay, done. This is also clear to all of us. As also, if each strand from a DNA, let us call it as the parent DNA, act as a template for the synthesis of the new strand. The two double-stranded DNA, let us call them as the daughter DNA, thus produced would be identical to the parent DNA molecule. Because of this, the genetic implication of the structure of DNA became very clear. That is, if I take this previous one, okay, if I take this as the parent, and if this is going to act as a template, I am going to get a daughter DNA which is going to be complementary to that. Same way, if let's take this 2 as the parent, both are the parent. If I take this separately and make them the template also, the strands I am going to get are going to be still complementary and they are going to be similar. Yes or no, they are going to be similar. So, this is what they have mentioned. So far, good students? Yes? Properly clear, because we are going to the salient features. Okay, we are going to the salient features. Okay, the salient features of double helical structure of DNA are as follows. First one, it is made up of 2 polypeptide chain where the backbone is constituted by sugar and phosphate. We already know this, and the bases are going to project inwards so that they can stay. Okay, they can stay. Now, the 2 chains have anti-parallel polarity. That means, in the one end, if the 5 dash and 3 dash is there, in the other end, 3 dash and 5 dash is going to be there. That means, on the 5 dash and the phosphate is going to be empty, on the 3 dash and OH is going to be empty. Vice versa will happen in the other place. Okay, yeah, I'll give you a break. Okay, I'll give you a break. It means, if one chain has a polarity 5 dash to 3 dash, the other will have 3 dash to 5 dash, right? The bases in 2 strands are paired through hydrogen bonds. That is, that 2 pairing will form the base pairs. Okay, that 2 pairing will form the base pairs. And adenine forms 2 hydrogen bonds, while cytosine and guanine will form 3 hydrogen bonds. All this we discussed. Okay, so salient features are going to be super duper kutti things for you guys. Yes or no? Adenine forms 2 hydrogen bonds with thymine. Okay, with thymine. One second, students, these lights are hurting me. You guys want lunch break? Okay, chalo, I'll give you lunch break then. Is it better now? I might look a little dark, but too much light on the screen. Okay, as a result, always a purine comes opposite to the pyrimidine. This generates approximately uniform distance between the two strands of the helix. Now, this is a very important point. Okay, the two chains are coiled in a right-handed fashion. Okay, it is coiled in a right-handed fashion, and the pitch of the helix is 3.4 nanometer. A nanometer is 1 billionth of the meter, that is 10 to the power minus 9. Okay, and they are roughly 10 base pairs. That means in this, okay, this is a DNA strand. Like this one strand is there. In this one turn, okay, let's say this middle portion, in this one turn, you will be able to find 10 base pairs. Okay, 10 base pairs, and the pitch is going to be 3.4 nanometer. This is what they have mentioned. Okay, this is what they have mentioned. Okay, clear? Chalo. Now, consequently, the distance between a base pair in a helix is approximately 0.34 nanometer. Students, it's very simple, no? Pitch is 3.4 nanometer divided by 10 uses, sorry, is equal to, suddenly my tongue just twisted. Okay, so what is going to happen? They are telling the pitch 3.4 nanometer. Okay, in one turn. Okay, it's going to be 10 base pairs. So, they are asking how much will be the helix. Okay, that it is going to be what? 3.4 divided by 10, which is equal to 0.34. Okay, one turn, how much? Okay, one turn. Okay, break. Okay, I'll give you. My God, these guys are crying now. Okay, the plane of one base pair stacks over the other in a double helical. This, in addition to the hydrogen bond, confers the stability. That means the plane of one base pair, so one base pair over another base pair, over another base pair, over another base pair. Okay, that is going to confer the stability of helical structure. Chalo, break I'll give you people. Otherwise, you people will break my head. Okay. How long you want the break? Do you want a lunch break or you want a chuttu break? Kuti break, lunch break? Tongue twisted successfully it seems. Okay. Till now it is what time, students? 1:47, huh? Let's assume 1:50 only. Two 5 works. Done. Okay. 2:05 to 2:10 gap, please come back. Okay. 2:05 to 2:10, because by the time I step out only it will become 2 o'clock. In this gap, try to come out. Okay. Come back. Done. Yes. Okay, lunch break. Yeah, this I've given you 20 minutes break, so have lunch and come.
guys.
Oh, all of you are back. Very nice, very nice. 1 second. Yes. Time up. Oh my god. All clocks in my class. So much they are finding out. Ready? So far everything was good, clear, perfect, no? Now we'll continue, okay? Yes, chalo, let's continue. Okay. Target done. Yes, yes, I saw. Very nice, very nice. Okay, done. So students, salient features done. Now we have to learn about central dogma. So from here the chapter is actually very interesting. Okay, chapter is very interesting. No, I'll have lunch later guys, because I cannot go this fast and have in 15 minutes and all. I cannot even reach the restaurant near me. Okay, so I'll have later with Diksha ma'am. So yes, central dogma. So what is central dogma, ma'am? This entire chapter revolves around central dogma. If I tell, will you believe? Yes, ma'am, I will believe, right? That is, I have to make DNA to mRNA, mRNA to protein. That is my job. Now, protein is going to decide how this person is going to look, what are the characteristics, all this phenotypes, genotypes, phenotypes are going to be determined, genotypes are going to be determined by these proteins, right? Because they are going to code for a protein, and that coding is going to decide what is going to be the end product, okay? So yes, let's see what is this. So this is the central dogma, that is, DNA will become mRNA, and that process is simply called as transcription. Okay, that process is called as transcription, where a DNA can become mRNA. And if mRNA has to become protein, that process is called translation. And this protein will, that is, this protein is formed by combining many amino acids together. Okay, combining many amino acids together, they will form a polypeptide chain. Okay, polypeptide chain. Got it? All of you clear? Now, here they are telling, ma'am, one circle why are they going? That is, if DNA can form its own copies, right? DNA can form its own copies, we call it replication. Oh, okay, we can call it replication, and our DNA replicates in a semi-conservative manner. What manner, students? Semi-conservative manner. Clear to all of you? Yes or no? Sure. Thank you so much, Harshine. I will definitely do that. Yes. Okay, cytoplasm will come. Okay, we'll come to that. Yes, protein is our decider. Okay, this is clear now. This is the central dogma, and we are going to revolve this entire chapter on this basis. We will start with replication, then we'll go to transcription, translation, and then we will go understand more about proteins. Okay, now let's learn a very important concept, students: packaging of DNA. Imagine a 2.2 meter long, approximately 2.2 meter long of DNA. I want to keep it inside a small nucleus. Now, how will I do that? It's very difficult, no? Imagine a 10 minus 6 meter. Inside that, I have to keep this 2.2 meter long DNA. For that, now I have to find a different type of binding itself. I have to learn how I can take this long thread. Okay, let's say this is a long thread. If this comes out, we are lucky. Okay, see, this is a long thread. Okay, this big thread it is. Imagine this entire thread, I have to fit it into a small place. Now, what will I do? I will coil it. Okay, I will coil it, or I will wound it around something like a bead like structure, how we make the chains, no? Like this, I will wound this whole thing. Now you see this long thread, how they have kept it, they have made it this small. But once I remove this, it can maybe fill half of the studio. I can like draw a border for the studio, right? So like that. So same thing is going to happen. For that, we are going to use some people. But before that, let's see NCERT, what NCERT is taking. taken the distance between two consecutive base pairs as 0.34, that is 0.34 into 10 to the power minus meter. Nanometer, I converted it into meter. It is going to be this much. We already did this calculation as well. Yes or no? We did the calculation and we understood that. How we got that? If the length of DNA double helical in a typical mammalian cell is calculated by simply multiplying with the total number of base pair with the distance between two consecutive base pair, that is, students, this is the calculation: 6.6 into 10 to the power 9 base pair into 0.34 into 10 to the power minus 9 base pair. How much is it going to be? All of you do maths. All of you do maths quickly. Okay, simple maths, no? Maths is going to be 6.6 into 0.34, okay, because 10 to the power 9 and 10 to the power minus 9 will get cancelled, and if you do this you will get approximately 2.2 meters. So this 2.2 meters of DNA, I have to fit it inside a nucleus which is 10 to the power minus 6. So a length that is far greater than the dimension of a typical nucleus, that is approximately 10 to the power minus 6 meters. How is such a long polymer packed inside a cell? All of you tell me, how is such a long polymer packed inside a cell? We were like, hey, don't worry, man. Like how Gopika Ma'am is here, no fear. Same way, we will bring some histones from somewhere. Okay, we are going to bring a set of proteins, and that proteins are going to wound around this long 2.2 meter of DNA, and we are going to keep it inside the nucleus. So let's see that process. See. If the length of the DNA is 1.36 mm, can you calculate the number of base pairs in E. coli? Okay, they have given you the length of DNA in E. coli and they have asked you to find how many base pairs are there. Okay, so here students, just the same formula like how we did 6.6 into 10 to the power 0.34. You will have to know the E. coli. We already know E. coli is 4.6 into 10 to the power 6. Right? Just do the calculation, and here they have given. So I'll tell you how to do this. Okay, so students, what do we have? Let's say the length of the DNA is 1.36, okay, mm. You convert it into meter is equal to, now we do not know the distance between two base pairs, that we have to find. Okay, we know that this much base pairs are going to be there into x. Okay, I am going to consider this as x. So you have to find it this way, because same thing we did in human, no? Length of DNA we did not know, so we kept length of DNA as x, and we knew that the total haploid content, diploid content was 6.6 into 10 to the power 9 base pairs, and we knew the distance between two base pairs is going to be 0.34. So like that we found 2.2 meters. So here they have given us already this. With this you have to find this value. Understood how you should do maths? Okay, they'll be like, my god, what is this ma'am telling? Okay, okay, they'll be like, ma'am. So maths teacher has become x value you have to find. Once you find this x value, first you convert this into meters, then you find this, divide this, okay, it will go that's it this side, and you will get x. Okay, my god, so much maths. Okay, chalo. My maths teacher will be proud. In prokaryotes such as E. coli, though they do not have a defined nucleus, the DNA is not scattered throughout the cell. Even though we are telling that prokaryotes does not have a proper nuclear membrane, it's just like a something is there. It does not mean DNA is going everywhere, telling that, oh, I don't have a nucleus membrane, so I will just go everywhere. No, right? Your parents does not let you go anywhere, no, when they're not there. So they'll lock you and go. Same way. DNA being negatively charged is held with some proteins that have positive charge in the region termed as nucleoid. So the region in prokaryotes is going to be what? They're going to be nucleoid, right? In the nucleoid also, we are making sure that the DNA is going to be attached with a positively charged proteins. Positively charged proteins. Clear all of you? Yes or no? Now it is organized in large loops held by proteins. Okay. Are you sarcastic or are you people? I don't know what you people are telling. Okay. In eukaryotes, this organization is much more complex. Obviously, in eukaryotes, everything is complex. There is a set of positively charged basic proteins called histones. Okay. There is a set of positively charged basic proteins called histones. Students, tell me, why have we taken the positive charged proteins? Q, Y. Okay. Diksha ma'am's influence, students. I've learned so much Hindi recently. Okay. Tell me why we have taken positively charged proteins and not negatively charged proteins. Tell quickly. Quickly. Okay. Very good, ma'am. Because DNA is negative. If I run a gel electrophoresis, yes or no, I've taught you this in biotechnology. If I run a gel electrophoresis, my DNA is going to move towards the anode. Okay, it's going to move towards the positively charged because my DNA is negatively charged, and to keep them in place, I will need a positively charged protein. So, a protein acquires charge depending on the abundance of amino acids residue with charged side chains. Histones are rich in basic amino acids, so they're telling how the charge is going to come. The charge depends on which type of amino acid is going to bind. Okay, so histones are rich in, this question has come many times, so just remember, in basic amino acid residues called lysine and arginine. Okay, this question has come, you're going to remember this. Very easy, no? That histones are rich in lysine and arginine amino acid. Both the amino acid residue carry positive charges in their side chains. Histones are organized to form a unit of 8 structures. Ready to learn this? Yes. I've taught you this so many times, but still we are going to learn it once again. Okay, we're going to learn it once again. Okay, look here. You guys text in the Telegram and ask them to come back. This is why I don't give lunch break. After lunch, you know, they'll be like, okay, I'll sleep for some time. They think that they are, you know, that person who has reached up to the age of 70, retired and sitting at home. But after lunch break, they want to sleep. Okay, so packaging of DNA. Very simple, no, students. They have already mentioned your DNA is positively charged. Sorry, DNA is negatively charged. Okay. And that is because of your phosphate group, right? You have PO4 3 minus. DNA is negatively charged. So, we are going to use the help of histones. Okay, this is a set of proteins which are positively charged. Okay, a set of proteins which are positively charged. They are rich in amino acids called lysine and arginine. Now, how many histones are we going to use? We are going to use 4 histones mainly. Okay. 4 histones are going to be mainly used in the formation of the histone octamer. Which are the histones? 4 of them. H1. Are we using H1? No, ma'am, we will not use H1 right now. We will use H2A, H2B, H3, H4. Okay, so exactly this 4 histones we are going to use for making our histone octamer. Okay, histone octamer. So we will do, okay, one second. Okay, first we will draw our H2A, we will draw our H2, okay, H2B, okay, H2. This is your H2C and your, not H2C student, H3 and H4. Okay, in a flow it came. Okay, these are your octamers. Okay, this will form total. Now wait, we have one more task. Students, how can we form an octamer? Now I have taken only 4, no? So ma'am, they are going to come in double, right? 2 people are going to come. 2 people are going to come, and those 2 people are going to together form the octamer. So each content, right, each, let's say H2A, H2B, H3 and H4, 2 are going to be there and they are going to form our octamer, right? Yes ma'am, clear, very clear. So this is going to be H2A, H2B, H3 and H4. Now what are they going to do, students? They're not going to chumma sit like this. Okay, they are going to start taking the DNA. Okay, they are going to wound DNA around them. Let's say the DNA is like this. Okay, it is going around like this and it is coming around like this. So the entire wounding of DNA is going to happen. Now imagine if I am winding something. Okay, I'm rotating, no? Now I have to make sure that this does not move around. Now imagine I put a bead and I put a thread inside it. Obviously the bead is going to move that side and this side because the hole in between is going to be bigger than the thread. So it is going to move. I have to make sure that they do not move. So I will put a knot. Same way here also I cannot tie a knot, right? Because this is DNA structure, right? It's not making the chain. So here instead of tying a knot, what we are going to do? We will tell, histone will tell, hey, don't worry man, I have a solution. The solution is going to be, I have brought one more protein with me, and that protein is called as the linker protein. Okay, that protein is called the linker protein or the H1 protein. So histone octamer is made by 4 proteins, but we have total 5 histone proteins who are helping us in the formation or in the packaging. So H1 protein is going to make sure that everything stays in place and everything is going to be tight, and this protein is called as the H1 protein, or in other words it's called the linker proteins. Linker proteins. Clear to all of you? Yes or no? Easy peasy lemon squeezy. Right? Yes ma'am. Easy. Okay, linker protein. Done. Now this is done. Now they are telling me that, Gopika, you cannot just call this histone octamer. I told okay fine. Then what should I call it? So they are told that this is histone octamer. Okay, this is histone octamer without the DNA. Okay, without the DNA, I can call it histone octamer. Without DNA, I can call it histone octamer. Then I asked them, okay, then tell me another name. So they told me that if you want to call this, that is going to be histone octamer plus DNA is going to form a nucleosome. What are they going to form, students? Nucleosome. So this name is very important for you. Histone octamer plus DNA is going to be called nucleosome. Many nucleosomes, okay? Many nucleosomes are together going to form your chromatin fibers. Many nucleosomes. So let's say this is a histone. Okay, this is your DNA. Okay, this will continue like this. Okay, it will go like this. Okay, again it will continue and it will finally form the chromosome. Okay, so it will, like this it is going to happen. Clear to all of you? Yes or no? Okay, this is how it is going to happen. Okay, many nucleosomes together will form the chromatin fibers. Chromatin fibers will ultimately become your chromosomes with your sister chromatids, which you will study in your principles of inheritance. Okay, clear or not? Yes. Clear. Done? Yes ma'am. Okay, now if I view this, okay, if I view this under the microscope, I will get a bead on a string structure. What will I get? A bead on a string structure. If I see this under the microscope, I am going to get a bead on a string structure. This is all about your packaging. Okay, this is all about your packaging. Now here you just remember about your lysine and arginine. Okay, lysine and arginine. Clear? Beautiful, no? Beautiful board. Okay, so take these notes and definitely learn from this. Now students, they are telling me that in eukaryotes, okay, they are telling that in eukaryotes, when I want to do more complex work, okay, in eukaryotes, I have to use an additional set of proteins. Okay, for higher, condensation, I have to use higher set of proteins, and that proteins are called NHC. That is non-histone chromosomal protein. Non-histone chromosomal protein. Clear to all of you? Yes or no? non-histone chromosomal proteins. I will use it in eukaryotes when I need to do additional condensation. Okay, I have to do additional condensation. I will use this non-histone chromosomal protein. So, charges are clear, students. Negative charge is there, and that is also because of the presence of phosphate. Okay, presence of PO4 3 minus. Okay, presence of phosphate, and histones are positive charge. Proteins like histones are positively charged. I did not write that. They are positive charge, and that is also because of the presence of different types of amino acids. Clear? Everything is super clear? Yes? Okay. Evolution I have done, no dear. Okay, now we will just go quickly through NCERT. Will not take much time. Okay, will not take much time. We will do quickly through NCERT. See, the negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. Understood? You shouldn't call the histone octamer nucleosome. Only when the DNA is wrapped around the histone octamer, you are going to call it a nucleosome. Clear? Okay, ma'am, clear. A typical nucleosome contains 200 base pairs of DNA helix. How many base pairs? 200 base pairs. In one turn, we have only 10 base pairs. 200 base pairs are present in your nucleosomes. Nucleosome constitute the repeating unit of a structure in nucleus called chromatin. That is a thread-like stained colored bodies seen in the nucleus. The nucleosome in chromatin are seen as a bead on a string structure under the microscope. Okay, one diagram also is given for you, under the electron microscope. They've shown you the structure, that bead on a string structure. Nothing, students, it's very simple. Okay, don't be like, oh, what is this bead on a string structure? Like this. Okay, it will be like this. This is the bead on a string structure. Now, theoretically, how many such beads do you imagine are present in a mammalian cell? Okay, so you can just do that into 200 base pairs. Okay, you can just do the haploid content, diploid content into 200 base pairs. The bead on a string structure in a chromatin is packed to form chromatin fibers that are further coiled and condensed at the metaphase phase of the cell division to form the chromosomes. Now, in prophase, you will be able to see this. Okay, all of you, how many of you have done this experiment in your PU? In your PU, you have done this in the lab, and you have got scoldings from your teacher telling this is not prophase, this is some, some khichdi that you have found. Okay, you smash the root tips so heavily, or you have taken, I don't know, for different, like you usually do mitosis, no? We take for meiosis root tip, we take for mitosis onion peel. Remember that white transparent layer. Okay, so different colleges take different things. Okay, but I'm telling generally, students, in prophase, what do you see? You see a condensed structure like this under the microscope, right? By the time it becomes metaphase, okay, by the time it becomes metaphase, you will slowly start seeing the chromosomes. Again, they will not move correctly, but then you will slowly start seeing the chromosomes like this. Yes or no? Like this. Okay, in the anaphase, what will you see? You will see that they have split. Okay, the sister chromatids are split and they are moving towards the poles, and there is asterase, right, forming, spindle fibers forming. Okay, you will see like this. Okay, you will see like this, they have moved towards the pole. Like this structures you will see, right? Okay, like this, from the equator, they would have moved. Okay, they have moved. So, this is what they are telling you, that you have seen it. Okay, you have seen it. Okay, chalo. Now, this is clear, no? Chromatin at higher level. See, look here. The chromatin, okay, the chromatin at a higher level, the packaging of chromatin at higher levels require additional set of proteins that collectively are referred to as your NHC, which is non-histone chromosomal protein. In a typical nucleus, some regions of the chromatin are loosely packed. I have drawn this and showed you all of you last time. See, students, in a, let's say this is the entire chromosome. Okay, I am taking one portion of the chromosomes. Okay, this is your entire chromosome. Too big it became, no? Okay, wait, let's draw basic size. Okay, this is your one chromosome. In this chromosome, they are telling that there are some regions, okay, there are some regions which are tightly packed, and then there are regions which are loosely packed. Okay, again there are regions which are tightly packed. Remember? So this is what they are mentioning, that there are regions which are loosely packed, there are regions that are tightly packed. Depending on that, look here, they have names also for it. See, in a typical nucleus, some regions of the chromatin are loosely packed, okay, and stains lightly and are referred to as euchromatin, okay, euchromatin. So here this portion is going to be called the euchromatin. Euchromatin is loosely packed, okay, loosely packed. Students, because they are loosely packed, obviously they will stain lightly, okay, stains lightly. Then they are telling there are some regions which are tightly packed, okay, there are some regions that are tightly packed, that is going to be called your heterochromatins, okay, that is going to be called your heterochromatin, okay, heterochromatin, which is tightly packed, okay, tightly packed, and because they are tightly packed, they are going to be staining darkly, okay, they will stain dark. Got it? Now one more word is there, the transcriptionally active and transcriptionally inactive, that is euchromatin is going to be transcriptionally active, this one, okay, this is going to be transcriptionally active, and this is going to be transcriptionally inactive. Clear? Done? Easy? Understood? On the nucleus, there will be regions where there are darkly stained, lightly stained. Lightly stained is called euchromatin because they are lightly stained and loosely packed. Okay, because they are loosely packed, they are lightly stained. Okay, and they are transcriptionally active. The other one is going to be dark. So that is going to be heterochromatin. Easy peasy. All of you tell me. Shall we go to the first experiment, Griffith's experiment? Okay, I think today we can only do up to the experiment, students. More than that, I don't think we'll be able to do. Okay. Yes. Chalo. Okay, very nice. So, very easy, no? Griffith's experiment is like a baby experiment. So, we'll just go through it very simple. Okay, in 1928, Frederick Griffith, in a series of experiment, okay, he did many experiments, in a series of experiment with Streptococcus pneumoniae. Now, in the chat box, my students who have done human health and diseases with me is going to tell me what is Streptococcus pneumoniae going to cause? Tell me, answer is there in the question only. Okay, don't see. Okay, bacterium which causes pneumonia. Okay, bacterium which causes pneumonia. So, question is invalid. Witnessed a miraculous transformation in the bacteria. During the course of his experiment, a living organism had changed in physical form. That is, they are telling that he noticed that when he was doing many experiments, he noticed that Streptococcus pneumoniae at one point from being non-virulent, it became virulent, right? You already know the experiment, so I'm just telling it before really. When Streptococcus pneumoniae or the pneumococcus bacteria are grown on a culture plate, some produces smoothly shiny colonies, which is called the S strain, while the others produces rough colonies, which is called the R strain. Okay, this is because S strain bacteria have mucus or polysaccharide coat. So, if something has to be smooth, that means we have to do proper bordening of it, right? Proper bordening should be there. So, they are telling that this S strain is smooth strain, and that is because of the coating. Okay, because of the coating. That mucopolysaccharide coating or that polysaccharide coating is making it smooth. But now also people believe that polysaccharide or the mucopolysaccharide is the reason why smooth strain is virulent, because if you compare R strain and S strain, this is there in S strain which is not there in R strain. So, it could be the reason, right? It could be the reason. While R strain does not. Mice infected with S strain virulent die due to pneumonia infection, but mice infected with R strain will not die from pneumonia. Okay. This is the experiment. Okay, you think from Griffith's point of view and you tell what was shocking. See, when he injected S strain, we already knew that Streptococcus pneumonia had two strains. S strain was the smooth strain, R strain was the rough strain. Why am I calling S strain the smooth strain? Because of the presence of mucopolysaccharide. Why am I calling R strain? Because it is a rough strain. Okay, because it did not have a mucopolysaccharide. They are telling that S strain is virulent. Virulent means what? Disease causing. It has the ability to cause a disease. Okay, it has the ability to cause a disease. So it's telling that S strain is virulent. So whenever I inject S strain into the mice will die. Whenever I inject R strain into the mice will live. Problem sorted. Problem sorted. Okay. So whenever we injected S strain into the mice will die. Okay. Result 1. Second thing, when I injected R strain into the mice will live. Now he was like, okay, this is a very normal experiment. Now he thought, let me do something else. That is, Griffith was able to kill bacteria by heating them. He observed that the heat killed S strain bacteria. Okay, heat killed S strain bacteria when it was injected into the mice. The mice lived. That means what, students? Now why do we boil water? We boil water because of the fact that the germs in the water will die. Okay, germs in the water will die. Same thing he did here. He actually made this non-virulent for some time. Okay, he made it non-virulent. That means, see, every content of it did not die. Something got just suppressed that led the mice to live. But the same thing, he was like, okay, see, in this case also mice is living. In this case also mice is living. So he decided that I will do one thing. I will do joint venture. Okay, I will do a business only with these two people. I will do a joint venture. And he used S strain heat killed and R strain live. Okay, he did not heat kit or anything. He just used the live. And when he injected into the mice, students, shockingly the mice died. Now, if you were Griffith, you would have got shock. Right? Yes or no? If you were Griffith, you would have got shocked. Yes or no? Tell me quickly. He was like, see, I don't know what happened, but I know for a fact that from the S strain, okay, from the heat killed S strain, something got transformed to the R strain that killed the mice. So the thing that got transformed, I do not know anything about it. So he told that let me do one thing, I will call this the transforming principle. He called it what, students? The transforming principle. Obviously, if I tell that I found transforming principle, will you people keep quiet? No, you will also be like, wait, I also want to find. Like that, many other people came telling that I want to know exactly what transformed from the heat killed S strain to the R strain. Behind that, many scientists came. Okay, many people came, and then many people, one people was your biochemical. Okay, biochemical characteristic of your understanding what was exactly transformed. Okay, what was exactly transformed. Okay, now look here, biochemical characteristic of the transforming principle. So Griffith stopped it at transforming principle, telling that I know for a fact that something came from this heat killed S strain to R strain and made R strain virulent, but I do not know what is it. So I will basically call it the transforming principle. Then 3 other scientists came telling that we will find what is that, and that was none other than your Avery, MacLeod and McCarty. Okay, Avery, MacLeod and McCarty, they were like, we will try to find what exactly happened, and the experiment went for almost 10 years, that gap. During that gap, they did it. The genetic material was thought to be protein. They worked to determine the biochemical nature of transforming principle. So, if you read NCERT, you will not technically understand what it is, but first I will read, okay. See, they purified biochemicals. What are the biochemicals present in this S strain? It would be proteins, RNA, DNA. Yes or no? Lipids, carbohydrates, fats and all. We know that it cannot have this much ability also. Now, from the heat killed S cells, to see which one could transform the live R cell into S cell, they discovered that DNA alone from S bacteria caused R bacteria to become transformed. So they told off. They were like, hey, you know what? DNA is the transforming principle. That fellow only came from this S strain and made this Papa R strain virulent. Okay, sometime in our life also there'll be some people, no, they'll keep telling us, you know what, this person is not, and we will listen to that person and we'll be like, now I'll become the villain. Okay, but last what will happen, this fellow and that fellow will become best friends and you will become the only villain in the story, right? So that is what happened. So they are telling, you know what, DNA only jumped from S strain to R strain and made R strain all problematic. Okay, but the experiment pattern that they took was not convincing for many people. They were like, you know what, I'm not convinced. So many people told, I'm not convinced, then their experiment got rejected. But let's see what's their experiment. Okay, they also discovered that protein digesting enzyme. So they took 3 enzymes. What are the enzymes? First they took protease, because the biochemical that they extracted was protein, RNA and DNA. So they took first fellow telling that you will eat protein, no, I will take you. So they took protease. Same way they told that you will eat RNA, no, I'll take RNA. Same way for the DNA. So they took protease, RNase and DNase. Okay, and RNA digesting enzyme, they took RNase. Okay, and they noticed that that also did not affect. So the transforming substance was not protein or RNA. Digestion with the DNase, so they took third one was DNase, did inhibit transformation, suggesting that the DNA caused the transformation. They concluded that DNA is the hereditary material, but not all biologists were confirmed, because biologists, people cannot be convinced so easily. They were like, no, your experiment is not very convincing for me, so I'm not going to follow this experiment. Okay, now what did the experiment happen, that I'm going to tell you. Okay, because this experiment questions have come and experiment is not very clear. Okay, so then what they took is, they took 3 enzymes. Okay, 3 enzymes they took. Which were the 3 enzymes, ma'am? First they took protease, then they took RNase, then they took DNase. Now we know for a fact that protease will digest the protein. Yes or no? It will digest protein. Okay, this fellow will digest whom? RNA. And this fellow will digest DNA. Now what did they do, students? They took the same combination. Okay, they took heat killed S strain plus R strain combination, and they were trying to check the biochemical nature of this. Okay, they were trying to check the biochemical nature of this. So during the biochemical nature, they had already understood that we are going to take few things, but as of now only 3 things are mentioned. Now in this, first they added protease. First they added what? Protease. You tell me what would have happened to the mice. You people are crying for mice justice, no? Tell me what would have happened to the mice. Died or alive? It's going to live or not live? Tell me. Quickly. What is going to happen to the mice? Yo, these people are only crying for justice. They don't even bother about mice. Yes, correct. After that, tell me more about it. Ma'am, the mice will die, ma'am. Obviously, why? Why will the mice die? Because DNA is still active here. Yes or no? DNA is still active. Okay, this experiment happened. Second thing they took was, students, he took RNase. Okay, I'm writing in one setup only same thing. Okay, RNase he took. Here also, the mice died. Because why? DNA is still active. You did not kill the DNA. How can you tell that it is going to happen? Then they finally took DNase. They added DNase. When they took DNA, obviously the DNA got digested, so the mice lived. The mice lived. Okay, that is when they understood, oh my god, it is the DNA that is the transforming principle. Okay, the DNA is going to be the transforming principle. So they understood DNA is the genetic material. DNA is the genetic material. Clear to all of you? Yes or no? Thumbs up in the chat box if you all understood. Okay? Okay? Clear? So DNA is the genetic material. This was the experiment by Avery, MacLeod and McCarthy. Avery, I want all of you to share this link with your friends and let me know in the comments how you understood NCERT line by line. Okay? Yes or no? Will you all do that? Yes. Okay. Done. Understood. This was the experiment. Then students, obviously many people were not happy. Then next set of people came telling that, you know what, we have a better experiment idea. Who was that people? Hershey and Chase. Okay, Hershey and Chase. That is, Hershey and Chase was like, wait, the unequivocal proof, that means the one and only proof that DNA is the genetic material came from this experiment done by Hershey and Chase. Okay, this question has come as such, this statement has come as such. So you should remember this, okay? Hershey and Chase experiment, they worked with virus that infect bacteria called bacteriophages. So what is bacteriophages? They are the virus, okay? They are the virus who will eat bacteria, okay? Virus who eats bacteria or kills bacteria, no? Okay, virus who eats bacteria. The bacteriophage attaches to the bacteria and its genetic material will enter into the bacterial cell. Students, what is the biggest problem with virus? As soon as this fellow attaches with the host, no, he'll be like, oh, this feels like a home. So he will release the genetic material inside. Okay, there are some people, no, in our lives also where they'll come and they'll feel so comfortable that they will start talking non-stop and they'll tell about their childhood, they'll tell about their traumas, so much they will talk that we'll be like, wait, how do I even know this person? So like that, this virus is like very comfortable. The moment it sees an host, no, he's like, oh, all my problems are over. I'm going to put all my genetic information into it. Okay, not just my childhood traumas and adulthood. I'm going to put my whole genetic information. So same way, it will just release the genetic material into the E. coli. Now here we are taking the E. coli. The bacterial cell treats the viral genetic material as if it is own. This is why I told you. This is why I told you. Understood? Okay, as its own and subsequently manufactures more virus particles. It just feels like, oh, you're my own. Now I will do one thing, I will start one daycare also inside. I will start making more viruses inside. Okay. Hershey and Chase worked to discover whether it was the protein or a DNA from the virus that entered the bacteria, because they were sure it was not RNA, right? Because bacteriophage had DNA. Why will they think about RNA? Correct? Now, they grew some virus on a medium. This is very important. Okay, first what did they do? They're very smart. Okay, their experimental thing is very like proper. Okay, they grew some virus on the medium that contained radioactive phosphorus. So they did the experiment in such a way, students, that they took radioactive materials. They took phosphorus. Okay, they took phosphorus. Okay, they took P32. And they took sulfur. Okay, they took sulfur S35. Now, why they took this? It was very clear. Phosphorus is present in DNA, sulfur is present in protein. It's not present in DNA. So, this way what will happen? If I am getting a radioactive substance, I will exactly know is it DNA or protein. Understood? To make the difference very clear, they took a radioactive substance with phosphorus, they took a radioactive substances with sulfur. Okay, and they made sure that, okay, if this fellow is going to be there, that means I know it's a DNA. If this fellow is going to be there, I know it's a protein. Understood? So, they grew some viruses on a medium that contained radioactive phosphorus and some others on a medium that contained radioactive sulfur. Virus grown in the presence of radioactive phosphorus, look here carefully, contained radioactive DNA. Why? Because in DNA phosphorus is there, in protein phosphorus is not there. Okay, and not radioactive protein, because DNA contains phosphorus but protein does not. Clear or not? The statement they can ask you like statement based, right or wrong, assertion, reason, anything can come. So just be careful about this. But protein does not. Similarly, virus grown on radioactive sulfur contained radioactive protein, because of the same reason that virus grown on radioactive sulfur contained radioactive protein but not radioactive DNA, because DNA does not contain sulfur. Okay, yes. You started calling them uncle, huh? Why? Because they found DNA as the genetic material. See, you people are also same. You are like that bacteriophage, you are like virus. 2 minutes you study about them, you will start calling them uncle, auntie, after some time mother, father, everything you will call them. Okay, so comfortable, you think it's your own, right? Very nice. Now, radioactive phages were allowed to attach to E. coli bacteria. Then as the infection proceeded, the viral coats are removed. See, the viral coats were removed from the bacteria by the help of agitation, that is, students, with the help of blunder. not blunder, blender. Okay, the virus, that outside coat was removed. Okay, so first job was infection. Okay, first step was infection. Then second step was blending or agitation. The virus particles were separated from the bacteria by spinning them in a centrifuge. Okay, bacteria which was affected with virus that had radioactive DNA was radioactive. So look here, virus that had radioactive DNA were radioactive, indicating that DNA was the material that passed from the virus to the bacteria. Okay, virus to the bacteria. Bacteria were infected with virus that had radioactive protein, were not radioactive. So don't worry, I will teach you this with the help of a diagram. Okay, look here. Okay, thank God. Okay, now look here. So that is your bacteriophage. Okay, that is your bacteriophage. Who is your person who is going to affect or infect the bacteria? Okay, infect the bacteria. Got it? It is going to infect the bacteria, and here you can see one set is there, here one set is there. Here who is there? S35 is there. Here P32 is there. Okay, P32 is there. Now first job was infection. So here you can see red color shows that protein code got radioactively labeled. Here red color in the DNA shows that DNA got radioactively labeled. Okay, first job was infection. So here what is going to happen? We are taking E. coli cells. Okay, the bacteria used is E. coli. Papa, this E. coli only everywhere. Okay, why you people not asking justice for E. coli? Why you are only talking about mice? Ask about E. coli also. So E. coli was taken, infection happened, bacteriophage was infecting the E. coli. Okay, this is the bacteriophage, it infected the E. coli. Now second job is what, students? We understood that the moment virus enters the host, it is going to release its genetic material, right? It is going to release GM, that is the genetic material, into the host. Okay, into the host. After that, we do not have to wait for longer time. We did blending, we did agitation. Okay, and because of that, in this blending process, protein coat got removed. Protein code removed. See, if protein was the substance that was the genetic material, or that was who was going to get transferred, then protein should have stayed. No, but protein did not stay. Protein is like, okay, my work is over, I am going. Then they did centrifugation. Why? Once you do centrifugation, you will be able to get two things. You will get a pellet and you will get a supernatant. You will get a pellet and you will get a supernatant. Pellet is the portion which is heavier. Okay, so students, if this is the centrifugation tube, okay, I think chemistry ma'am would have taught you this. This is the centrifugation tube. In this, you will get two things. Okay, you will get supernatant. Okay, this is the supernatant. This is the lighter particle. Okay, and you will get a pellet. You will get a pellet which is like this, attached to the bottom of the tube, which is going to be heavier. So will genetic material be heavier or lighter? Obviously genetic material is going to be heavier. Okay, genetic material is going to be heavier. So genetic material will get settled in the pellet, and the pellet indicates the radioactivity also. So it was clear that genetic material was DNA. Okay, radioactivity was seen in the pellet. In the supernatant, we saw the here in this side we saw supernatant, protein coat was there, radioactivity was seen here, okay, which is of no use. So here you see what happened after centrifugation, students. Here no radioactivity detected in the cell. That means protein is not entering the cell also. Then how can I tell that you are the genetic material? Will people agree? Yes or no? They will not agree. Okay, yes. Okay, now those who joined right now, quickly mark your attendance. Okay, like the video as well, because I want to know how many of you were there with me live and how many of you enjoyed the video. Now students, radioactivity detected in the supernatant. Okay, radioactivity detected in the supernatant. That means protein coat was found in the supernatant. Here radioactivity detected in the cell. That means what? Cell who entered, that fellow is going to be the transforming principle. So DNA entered into the cell. Radioactivity was seen inside the cell. It was seen in the pellet. No radioactivity detected in the supernatant. Is this clear, all of you? Tell me. Is this clear? Yes. Very nice. Very nice. Okay. This is your experiment. Okay. This is your experiment. Clear? Clear, clear, clear. Okay, next topic. Energy is there. Another one, 15-20 minutes. Okay, we will stop this class. Okay, 15-20 minutes. From replication also I'll try teaching you a little bit. Okay, from replication I will start in the next class, or from machineries I'll start in the next class. Okay, fine. Okay. 1 second, students. This mic has some. Today is a technical error day. So many technical issues in one day. Okay, chalo. Okay, I'll share the notes on Telegram. Okay, don't worry. I'll share the notes on Telegram. And I don't know how many of you saw the KCET video. If you want that notes also, just let me know. I will share it on my Telegram channel. Okay, I had written sexual reproduction in flowering plants. I have written handwritten notes. Okay, I have taken some specific topics and I have written. If you found that notes very helpful, just let me know now itself. Okay, you can go check V Karnataka KCET channel. I have done a video yesterday, and if you found that notes helpful, you can just tell me. I will share that notes also. Okay, come on. Can you recall two chemical differences between DNA and RNA? That is, students, if I want to be the MLA or if I want to be the Prime Minister. Diksha ma'am came at the wrong time. Diksha ma'am is like, how can you be the Prime Minister? Yeah, come. Students, MVSAT exam is starting. It started from 10th. It is going to finish at 20th. Please register now. The link is in the description box. Ma'am, tell about MVSAT.
So how many of you have actually enrolled to the MVSAT? Everybody. Have they enrolled or not?
Huh?
Let us see how many of they. Some of the students have actually given the test as well.
Yeah. Many of our daily motivated students have already.
Yeah. They're
Okay.
already there.
Yes.
Yes, Diksha ma'am. Hi, Diksha ma'am. Hello. So, okay, ma'am, how many hours you are teaching? It's been how many hours?
It's been only 2 hours.
Okay, you started the class at 12. Is it?
12:30.
12:30. Okay. I thought at from 10 a.m. ma'am is just
ma'am.
teaching.
I am
What
not
happened?
Diksha ma'am's. I don't have Diksha ma'am's the ATP. energy
No, the
should-
syllabus is too much for chemistry, ma'am.
If I was a chemistry teacher, we both would have full done 3 hours. Ma'am, another 15 minutes I'll be done.
Okay.
You will be waiting? Yes, ma'am will be waiting because ma'am hasn't had lunch. After some time, she'll eat me only. She'll tell, what are you doing? Okay? Yes. Very nice. Students, MVSAT exam is happening in the Vedantu app itself. Okay, on the Wave platform itself, you can see it. So what you have to do is just go down, look at the description box, first link. Register now for scholarship exam. Click on that link. I'll show you at the end of the video. If I click now, everything will go. So click on that link and you register. Put your phone number. Automatically, you will get everything. Okay. Chalo, very nice. Diksha ma'am and Mintu ma'am is together. So students, as I told you, if I want to be the Prime Minister, okay, it's a very funny topic. But imagine if I want to be the MLA of some place, or I want to stand for something, election, okay. Now what will I do? They'll give me a list of eligibility criteria, like you should be physically well, you should be mentally well, you shouldn't have any, record, any crime record shouldn't be there, you should be at this much age, you should have this political background, something like that. They'll give me some eligibility criteria, okay. I have not stood, so I don't know, okay. Results will come after the exam gets over. That means after 20th, checking will happen. After that, you will get the results, okay. Chalo. So it's a scholarship exam. You will get a lot of prizes. If you're for participation, you will get free study materials as well. Okay, results, everything I will announce it on the channel. Don't worry. So students, if I want to stand, okay, they will tell me that Gopika, you should have this eligibility criteria. I will tell, okay, fine, I have the eligibility criteria. I will tell and I will stand for the election. Same way, if DNA has to be the genetic material for most of the living organism, DNA should go to a criteria test. RNA also should go through a criteria test. So today you are going to learn what are the criteria to be the genetic material. Like if I want to be the genetic material, what are the criteria that I should follow? DNA and RNA is going to tell. See, if a molecule that can act as a genetic material must fulfill the following criteria. That is, it should be able to generate its replica. That means students, it should be able to undergo replication. But luckily in this, students, both of them are scoring marks. Are you with DNA or RNA? So we have a DNA batch running on paid batch, and we have an RNA batch which is the YouTube batch. So you guys tell me, are you with RNA or DNA? Depending on that, we have to see the score. So students, luckily both RNA and DNA has scored here because both of them will undergo replication. Okay, both of them will undergo replication, 1 point each. Okay, it should be stable chemically and structurally. That is not there. Actually, here also it's not there. My RNA batch students are not that regular. Okay, but my DNA batch students are very regular. So they should be stable chemically and structurally. So that stability is lacking here also. Here students, DNA scored. DNA 1 full score it has taken because the absence of oxygen made DNA so less reactive compared to RNA. RNA is like, I'll react there also, I'll react here also, I'll react there also. Why? Because that extra oxygen is there. Extra oxygen is there. Okay. It should provide scope for slow changes. See, both of them, both RNA and DNA is doing mutation because mutation is necessary for variation. So both RNA and DNA, both of them are going to do mutation, but RNA will do slow. Okay, sorry, DNA will do slow, RNA will do fast. Okay, like my students, they will like fast, fast, fast. Okay, they will like fast, fast, fast. Now, my RNA students like fast, they will comment fast also, right? DNA students are little slow in that same way. Here, that are required for evolution. It should be able to express itself in the form of Mendelian characters. That means it should be able to express in the characters mentioned by Mendelian, and it should also be able to show whatever phenotype, genotype, whatever it is, they have to express themselves. Okay? Yes. Now, here, let's see what makes it so different. Right, what makes it so different? Ready? Chalo. Okay, listen students. So, as I told you, first step is replication. First step is replication. Yes or no? Ma'am, first step is replication. Okay, sorted ma'am, clear. Replication, both of them are doing. In that, your DNA, okay, in that your DNA is going to do semi-conservative replication. Okay, semi-conservative replication, which I'll teach you later. RNA also is going to do replication. Okay, both are going to do replication. First point, both of them scored. Second point is going to be, they have to do mutation. Okay, mutation or slow changes. Slow changes. Here one problem is that. The problem is that DNA and RNA does mutation. DNA and RNA does mutation. Both of them does mutation. But what is the problem? DNA will do slow mutation. Students, do you want to sleep as a human and wake up as a penguin? No, right? We don't want that fast mutation also, right? So here RNA will do fast mutation. RNA will do fast mutation. Clear? Okay, chalo. Next is going to be the next point. What is the next point? They have to be structurally and chemically stable. Structurally and chemically stable. Correct? Wait a minute. Okay, they have to be structurally and chemically stable, students. That means what? That means, ma'am, they shouldn't react with anyone and everyone. They should have some solid, like, like you shouldn't go everywhere, right? You should be like, you should have that thing that I am very stable. I will not do any of this, right? Same way here, what is going to happen, students? DNA, the ribose sugar, right? The ribose sugar here, the second carbon, what has happened? The H group in the second carbon has made DNA more stable. So, the absence, I am writing all the points here, okay? One big paragraph is given in your NCERT. I am going to just write 3 points and make the work easy for you, okay? Absence of oxygen, okay? Absence of that oxy group is making it more stable. Presence of, okay? Presence of thymine instead of uracil is making DNA more stable. So these are few reasons why DNA is more stable than RNA. Okay, DNA is more stable than RNA, but RNA, because of the presence, okay, RNA, because of the presence of that OH in the second carbon, this OH, it has become highly reactive. Okay, highly reactive. Clear? Done. Ma'am, last point. What is the last point, guys? Last point is going to be that they have to be active in expressing your Mendelian characters. Expressing Mendelian characters. Okay, that also both of them will do, but again DNA does it better. Okay, GM plants. What do you mean genetically modified plants, sir? Yes, clear? This is your criteria, or in your textbook it's given properties. You can remember properties or you can remember criteria. Okay, these are your properties for being the genetic material. Understood? If they go through all these properties and if they approve of all these properties, then only you can be the genetic material. Okay, chumma today if you come and stand and tell I want to be the genetic material, they will not make you the genetic material. Okay, one more kutti topic we'll do, students, then next class I will start with semi-conservative replication. Ready? Okay, all of you can stay for that much time? Yes or no? Because I cannot take this chapter very continuously, because your brain will not brain after some time. Okay, I don't want your brain to stop braining also. Okay, chalo. One small topic, after that we'll be done. Okay, semi-conservative replication. Students, as you know, in the central dogma, we learned that DNA, okay, DNA can replicate. Okay, it will go through replication. It will go through replication, and this replication, I told you, it will happen in a semi-conservative manner. Okay, semi-conservative manner. That semi-conservative manner I am going to explain you with a very daily example, and I think all of you already know this example by now. Semi-conservative replication. Semi-conservative replication. What is semi-conservative? So, conservative replication, students. Let's take, this is the parent DNA. Okay, look like one braid, but it's parent DNA. Okay, parent DNA. So, for replication to happen, we need a template. Always remember, we need a template. I have already spoke about this in the starting of the class. No, template should be there. This template, with the help of the template, we are going to form the daughter DNA. Okay, daughter DNA. So, I am going to take the parents as this template. The parents were already formed, so I will take them as the template. No problem. So, each of the strand is going to act as a template. Let's say we separate them. Okay, we separate them. This is the template 1. Okay, let's say this is the mother. Okay, and this is going to be the father. Okay, this is template 2. Now, what happens normally in your home? I have always told this example to all of you. In your home, what happens, students? Let's say your siblings are fighting. If you both are fighting, what will happen? Mom will take one person's side, father will take one person's side. Okay, usually father will take younger sibling side and mother will take elder sibling side. So they will be like, oh, they will keep telling all this. Suddenly, after some time, you will notice that somewhere out of the blue, okay, they were fighting actually. That means we both are fighting, siblings were fighting. In between, father will tell one point about mother. You did this that day. Mother will tell you did this that day. Suddenly, it will become their fight. Now, initially the target was to solve our fight, but after some time what will happen? It will become their fight. So, this both who tell parent and daughter, that is mother and elder daughter will be together, father and younger daughter will be together. Now, it will become an entire family's fight. Okay, now area people have to involve. Okay, after some time police have to involve. It will become a bigger fight. Why I told this example, you will understand. Let's say this is the parent. Okay, parent will form a template. Yes or no? Parent will form a template. That is going to be your daughter DNA. Okay, it's going to be D1, I am writing daughter DNA, the newly formed DNA. Okay, same way this fellow also, that is this template also will form the daughter DNA. Okay, daughter DNA. Now, students, instead of parents going back together and the daughter DNA is coming back together, what is going to happen? they will stay together. Why? Because fight happened. No, this two will stay together. So this kind of replication is called semi-conservative replication. That means if I label these strands, let's say I'm labeling this according to your experiment N15, okay, N15 and N14. Okay, this will also be N15, N14. Understood? They will become hybrid. Instead of going back to the actual form, they will form hybrid. They will form hybrid. This is why it is called semi-conservative replication. Why is it called semi? Because one parent, one daughter. One parent, one daughter. It is forming hybrids. Clear all of you? Yes or no? Clear? This has happened in your home, I believe. Right? It happens in everyone's home. This is called as semi-conservative. This is called as semi-conservative, where we form hybrid. Okay, where we form hybrid. Clear? Okay, because technically what happens, students, in conservative, okay, technically if you see, normal replication, what happens is, this is the parent. Okay, this is the parent. We separated them to form the template. This is the template 1, template 2. After forming the daughter DNAs, okay, this is the daughter DNA, this is the daughter DNA. Automatically, these people will come back together. Okay, that is, the parent DNAs are going to come back together. Okay, and the daughter DNAs are going to come back together. Okay, but in our DNA, okay, so did the color change suddenly? Why do I look blue? So many colors today. Okay, so what is going to happen? No, it looks correct. Huh? I don't know. Yeah, that is very normal, Tanuja. Tell me about it. Okay, tell me about it. Suddenly they will, the worst part for you people is, now for me it is little better, but when I was back in school or colleges, same like how you guys were, suddenly they'll talk about my marks. That time I'll be like, oh no, it was actually they are both fight, but suddenly it will become towards me. Now everyone in the family will look towards me. Why is your marks like this? Okay, what are you planning in your life to do? So that will happen for all of you, because you are in your NEET preparation. Suddenly they both will be fighting. Somewhere one, financial topic will come, or some topic related to you will come, and suddenly they'll be like, what are you doing? When are you planning to, earn money so that I do not have to work and they do not have to work? So that conversation will suddenly come. So when everything turns towards you, that's when you get scared. I'm like, oh God, I was not even a part of the fight. I was sitting in one corner and doing my thing. Suddenly they both are fighting and they will look towards you. Yes or no? So that is the biggest problem for now, all of you, I know. Okay, see students, I know I have gone through all this, yaar. Adolescence was beautiful, no? That's why. So this is the parent, okay. This is the template formation. So with the help of template, okay, we will form the daughter DNA. If you understand this, next class will be easier for me also, for you also, because next class experiment I will teach you directly. From daughter DNA, this will go back to the conservative method. So this is conservative, where the parents are together. That means they came back because they are complementary. They came back and daughter will come back. But normally in all of our house, what will happen? Semi-conservative will happen. Same thing happens in our DNA as well. Okay. Christ University. Like you are asking in real, or like because I'm understanding this? Clear all of you? Okay. GM University. You people have made me GM artist, GM University, GM plants. I feel like we'll start a company by itself with lot of franchisees. Okay. You people are making me rich through chats only. Done. So next we have this experiment which we will do in the coming classes, and we will have enzyme machinery. Next class I think we'll have only experiments. Okay. Transcription, translation. Again HGP, fingerprinting will be there. Okay. Yes. So main chunk is over, students. Actually main chunk means the main portion that you people usually get confused is over. Now we have small portions which we can aram se do. Don't even take tension. This is revision. I'll have enough time. Okay. Right. Yes. Okay. 1 second. Let me help you people in one way. Students, all these notes you'll get it on Vedantu NEET English Telegram. Okay. And also I want to show you people one thing. Okay, this is your chats. Okay, let me see how many of you marked your attendance. I'm going to check the attendance, and if you people haven't marked, okay, now it's showing 40. Okay, students, see, look here. If you click on this, no, you will get the notes, because many students have been asking me notes, no. So if you go here, you will get NCRT line, you will get Navratri series. So here, whichever notes are not updated, I will update it soon. But here you can see most of the notes are here, okay. Evolution, I will add it here, and sexual reproduction I have already given. I think they would have added here, okay. They would have added here, okay. See here also, all the notes links are there, and the notes are here, okay. Notes are here. See, all these are note links. Just enter your phone number and you will get the notes. Okay, enter your phone number and you will get the notes. Same way for students who are asking me, one second, yeah. Okay, asking me about the MVSAT, just go click on this link, enter your phone number, press on submit, you will get an OTP. After you submit the OTP, you will get to know how it is going to happen. See, you can see here, the dates are going to be from 10th to 20th October. That means in 3 days MVSAT will get over in online mode, but one more day it will happen on the offline, that is on 27th October, up to 100% scholarship, and for participation you will get free study materials, okay. So yes, do join, okay, because many of you were asking, I showed it again, okay, otherwise I have already mentioned about it. Yes, so NEET treasure box also, don't forget, I will check the notes and I'll update you. Most of the notes are here, students, because again, same RNA notes, almost everything is updated. Only Navratri series, this is missing, that I will update it. I think they might have added it here, okay, they might have added it here. Okay, so I've sent the notes, sexual reproduction and this one, okay. Chalo, so I hope the session was really helpful. Let me also see your chats, okay. Just let me know in the comments if the session was helpful. And yes, okay, so I've taken it very slow, I have taught you in detail. So do let me know in the comments how you found the session, and definitely like the video so more students can reach students, because we are not asking this likes for us, we do not get that likes or anything like that, but the likes will indicate many students that there is a teacher like this and she is able to help so many students. Only you can make me reach to many other students, right? They will be like, okay, there is a ma'am who teaches in simpler way, I would love to. It will help actually many students, and I want all the students to be helped and many students to score well also in their biology exam, okay. So thank you so much for joining, see you all in next class, and yes, definitely do your part of the work, and I will definitely continue with the next thing, okay. So see you all, tata, bye bye.